Well, welcome and good day to everyone. This is the fifth and final session of the 2026 Spring Series on magnetic fields and biology, or magnetobiology. And as we have done in the past, Betony is going to summarize in grand fashion the talks that we've had from our speakers, and we have a number of speakers on today. And then that will be followed by a roundup presentation by Alistair. So let's go with Betony. Betony, let's kick off.
Thank you very much, Geoffrey. It is a real pleasure to be able to participate in another roundtable session, which always proved to have really interesting discussions. My job is always a fairly impossible one, choosing just 2 slides from each of the presentations over the course of the series. So I often end up choosing just the most general slides. And if I've left out anything integral, please let us know in the discussion. And thank you very much to the presenters for their abstracts, which I've used directly in this summary. So the theme for this series was magnetic fields in biology, as Geoffrey said. Magnetic fields are increasingly being recognized as crucial to biological processes, from bird navigation to human cellular functions. So in the series, we explored this magnetobiology field, including the underlying mechanisms, the evidence for magnetic field effects in plants, animals, and humans, and the potential for medicine. For those of you who couldn't make it to all of the talks, videos of each of the sessions are available on our YouTube channel, and I really encourage you to watch them, as a lot of the fascinating details the speakers presented will not be included in my recap today. That said, I will now go through each of the sessions and recap some of the major points that the different speakers raised. So we started the series with a focus on what physical mechanisms might underpin magnetic field effects in biology. While there are a number of different ways in which biological organisms might be sensitive to magnetic fields, including magnetic materials such as magnetite, our main focus in this session was the radical pair mechanism. Professor Jonathan Woodward gave us an excellent introduction to radical pair theory and how magnetic fields can modulate the rate or outcome of critical biochemical reactions. Classical theories of chemical thermodynamics and kinetics predict no measurable effects from weak to moderate magnetic fields. And this is because, despite the fact that unpaired electrons are inherently magnetic, their interaction with external magnetic fields is typically very small compared to thermal energy at room temperature. But the radical pair mechanism, which was first developed in the 1960s as spin chemistry became an established research field, provides a framework for how reactions involving these short-lived intermediates, which are known as spin-correlated radical pairs, can remain sensitive to even very weak magnetic fields, including the Earth's magnetic field. And specifically, this sensitivity arises because of the electron spin selectivity of radical pairs. Jonathan then detailed how the radical pair mechanism produces a distinctive dose-response curve, and he characterized these by 3 critical regions. Zero magnetic field, where mixing between spin states is inefficient due to symmetry restrictions on the total angular momentum of the spin states. Weak magnetic fields, where the applied field breaks this symmetry and increases mixing between spin states. And high magnetic field, where as the intensity of the field increases, certain non-reactive states become isolated in energy. So this makes mixing inefficient once more. Once he'd outlined the theoretical mechanism by which magnetic fields modulate chemical reactions, and I really recommend going to the Guy Foundation YouTube channel and watching a previous tutorial that he gave on the radical pair mechanism, which includes even more detail and is really instructive. Jonathan presented his own original research into how radical pair effects might function in real biological organisms, such as human cancer cells and bacteria. His experimental method utilizes real-time autofluorescence and how this responds to specific magnetic fields. And the results are really fascinating, and I think he mentioned that the paper would be out shortly. Very briefly, the studies show clear responses to modulated magnetic fields, including giant effects of over 10% on bacterial autofluorescence intensity. And even more interestingly, these responses fluctuate according to cellular stress and the presence of glucose. Session 1 concluded with a presentation by Dr. Brian Ross, who also took us through some of the theoretical principles underlying the radical pair mechanism and the intriguing experimental evidence that magnetic fields modulate processes in diverse organisms, from tadpoles to killifish to E. coli. He then went on to discuss the magnetic properties of biological materials, specifically proteins. At the molecular level, as demonstrated by Jonathan, the fluorescence activity of key flavor proteins can be changed by weak magnetic fields. Brian then went on to outline how their work builds on this foundation by applying methods from the field of technological quantum sensing to further investigate this magnetosensitivity. And in particular, the aim is to measure how long coherent spin superpositions can be maintained in the warm, wet environment of a living cell. Brian then discussed how the first step of such a project is to identify a suitable magnetosensitive protein that exhibits a strong fluorescence signal with high magnetosensitivity arising from the radical pair mechanism. And the protein maglev2 is an engineered flavor protein designed to produce large changes in fluorescence intensity in response to weak magnetic fields. He outlined how the magnitude of this fluorescence changes as a function of the strength of an externally applied magnetic field in E. coli colonies that have been engineered to express maglev2. And interestingly, in the context of the theory that we just heard about, this relationship is non-monotonic at low field strength and plateaus at higher field strength. And both of these observations are consistent with theoretical models of the radical pair mechanism, as we heard from Jonathan, who outlined those different relevant regimes of field strength. Moving on to session 2, the focus changed from theory to therapeutic practice. The session was introduced by Dr. Rees-Mould, but I will also include here the introduction given by Dr. Sinayskiy Gael in the next session, as both Rees and Sinayskiy discussed the Guy Foundation's own research into hypermagnetic fields in biology, particularly in the context of long-distance spaceflight becoming increasingly realistic. So using mu metal chambers that block static magnetic fields, Rees and Sinayskiy study the effects of hypermagnetic fields on a variety of cell line models. And preliminary data suggests that hypermagnetic field exposure alters immune cell function and stress response, cartilage cell morphology and muscle cell proliferation, as well as mitochondrial metabolism. I haven't included slides of the results explicitly as they are in the process of being officially submitted, but keep an eye out for the forthcoming papers. So after the introduction, session 2 turned its attention to medical research, and the first speaker of the session, Dr. Margit Egg, summarized the main published findings of their research group, which concerns the cellular effects of therapeutic nuclear magnetic resonance, or TNMR. TNMR consists of the application of a weak electromagnetic field, which is designed to generate water-proton resonance conditions. The method combines a low-intensity sweep field with a perpendicular radio frequency component that is set to the Larmor frequency of hydrogen. And clinically, TNMR is already used in musculoskeletal conditions such as osteoarthritis, osteoporosis, and wound healing. Margit also outlined which biological processes are relevant to their therapeutic NMR research, which includes the astronomical day and night cycle, low oxygen environments that are realistic in cartilage and bone-associated tissues and healing contexts, and mitochondrial redox signaling as a determinant of cellular adaptation. And previous research has shown that TNMR modulates cellular clock gene oscillations, indicating that responsiveness depends on circadian redox state. Margit then discussed therapeutic field effects, specifically in the context of intermittent hypoxia and acute low oxygen exposure. These conditions were modeled experimentally as 6 hours of TNMR treatment at 1% oxygen. And under these conditions, TNMR shifted cellular metabolism away from glycolysis, including reduced lactate production, while mitochondrial respiration remained preserved during reoxygenation. And magnetic field responses were also shown to involve compartment-specific reactive oxygen species signatures, with superoxide emerging as a central mediator rather than non-specific oxidative stress signal. And Margit concluded that these findings identify that timing, oxygen availability, redox state, and mitochondria are major determinants of therapeutic responsiveness. Session 2 concluded with a presentation by Professor Martin Sharp, who described how they have developed a way of generating rotating magnetic fields using a device they call an oncoscillator. The device uses the rotation of a neodymium magnet to generate spinning oscillating magnetic fields, or SOMF. Martin detailed how this oncomagnetic therapy exploits quantum effects to generate triplets or spin-forbidden states in paired biradical redox centers. And he suggested that one of the major targets of these spinning oscillating fields is mitochondrial complex succinate dehydrogenase. Within this complex, electron flux is inhibited, and during this inhibition, electrons are diverted into molecular oxygen, which generates ROS, reactive oxygen species. He then discussed how they have observed profound effects of these spinning oscillating magnetic fields on cancer cells in vitro, in vivo, and in end-stage glioma patients. Martin explained how diffuse intrinsic pontine glioma, which is now called diffuse midline glioma, or DMG, comprise approximately half of all pediatric high-grade gliomas, and these tumors are universally lethal and typically inoperable. He went on to discuss a case study that they conducted using oncomagnetic treatment. The 28-year-old woman in question had undergone partial surgical excision and radiotherapy of a DMG. Magnetic field treatment, which was initiated after the first recurrence of the tumor, was well tolerated for more than 694 days by the patient, and there was near complete regression of the imaged tumor at 145 days, with symptomatic relief, and a partial regression at 554 days, after an apparent progression at 518 days. Unfortunately, on day 694, treatment had to be stopped due to an unrelated cause. The patient required a hospital admission due to injuries from a fall and disease progression, which led to her death. Martin concluded that the aim is to introduce this effective, non-toxic and non-invasive wearable device-based therapy into clinical practice for the treatment of these deadly gliomas. The third session focused more directly on experimental results detailing how magnetic fields interact with human and animal models. Professor Wendy Bean discussed how research into the effects of magnetic fields on living systems has a long history, but the existing literature on human and animal model system data often features contradictory results, which has made it challenging to identify any underlying foundational cellular principles, which now should also take quantum effects into account. Wendy explains some of the factors that contribute to this complication or complicate a clear picture of magnetobiology. First, there are several biologically relevant areas where biological systems and magnetic fields interact, from magnetoreception to bioelectromagnetic communication between cells. There are also numerous types of magnetic field exposure, of which static, weak magnetic fields and extremely low frequency electromagnetic fields are the most commonly studied. Wendy then went on to explain how these problems persist even if research is narrowed to a specific area. For example, in her team's research on external static weak magnetic field exposure during planarian regeneration, the cellular and molecular magnetic field effects cover a range of signaling mechanisms. These include changes in cytoskeleton dynamics, membrane voltage and ion flux, and the generation of reactive oxygen species. And then to further complicate things, weak magnetic field effects vary by specific organ system, as cellular responses are context or tissue dependent and depend strongly on field strength. She outlined how their data show that planarian cells respond in a field-dependent manner to weak magnetic fields of less than a millitesla due to changes in superoxide levels at the wound site. So 500 microtesla exposures increase, while 200 microtesla exposure inhibit these ROS levels, and then subsequent gene expression, stem cell proliferation, and new tissue growth. Their work also reveals that many of these mechanisms are often integrated, and she detailed how they are currently investigating a bioelectric ROS signaling pathway that is targeted by weak magnetic fields. Wendy concluded by reiterating that this wide variety of magnetic field conditions and cellular responses make the initial synthesis of findings much more challenging, but at the same time, this very plurality promises future potential for a variety of specific therapies. In our fourth and final session, we completed the picture that was introduced in session 3 by expanding the scope of interest to include magnetic field effects in plants. Professor Massimo Maffei discussed how a primary focus of his research is on near-null magnetic fields, or NNM fields, which simulate environments lacking a magnetosphere, such as the Moon or Mars. So plants in NNM fields show suppressed growth, delayed flowering, and reduced biomass during the transition to reproductive stages. And this translates in a notable reduction in agricultural output. Massimo also detailed how simulated lunar magnetic fields disrupt photosynthetic carbon assimilation and affect ion homeostasis. And interestingly, these photosynthetic effects occur so rapidly, within seconds or minutes, that they are too fast to involve protein turnover or gene expression, which suggests direct physicochemical impacts on electron transport or membrane potential. He also addressed the possible mechanisms behind this response, such as the radical pair mechanism and the involvement of cryptochromes, which are blue light receptors where magnetic field effects occur specifically during the dark reoxidation period of the photocycle. In this phase, the magnetic field alters the reaction rate constant and can thus change the lifetime of the activated signaling state. And this then triggers significant hormonal impairment. Exposure to NNM fields also triggers a rapid reduction in root ion uptake and transport, affecting essential cations and anions. Massimo noted how this effect extends beyond hormones. Here, reduced magnetic fields trigger a transcriptomic and metabolomic shift, which is related to oxidative stress and circadian rhythms. NNM fields induce lower levels of ROS, like hydrogen peroxide, compared to geomagnetic field conditions. And this shift is paralleled by the regulation of ROS scavenging enzymes and a decrease in antioxidant polyphenols. The geomagnetic field also influences the circadian clock by altering the expression of clock-regulated genes, which impacts the timing of metabolic processes. And under NNM fields, plants show a biphasic dose response, which suggests a hormetic response. Massimo concluded by stating that understanding these responses is essential for any future space travel, and perhaps more immediately, terrestrial data from 1985 to 2015 shows a positive correlation between geomagnetic field intensity and crop yields, suggesting that magnetic field technology could be a sustainable, eco-friendly tool for enhancing global food security. With that, it's my pleasure to introduce our speaker for the session. Professor Alistair Nunn is Director of Science at the Guy Foundation and visiting professor in theoretical quantum bioenergetics at the Research Centre for Optimal Health at the University of Westminster. He's an active theoretician. He's worked in academia, the pharmaceutical industry, and medical communications. He's also published widely on mitochondria, quantum biology, medicine, phytocannabinoids, metabolic syndrome, cancer, space physiology, aging, and the origins of life. And just quickly, before I let Alistair take the stage, please look out for our 2026 Spring Series abstract proceedings, which will be available on the website from next month.
Okay, well, thanks very much. I think you all know me. I'm Alistair, and I work with the Foundation. And today I'm just going to try and look at some of the implications behind this. I think we've already covered quite a lot of it, but hopefully I can provide something which will just give a little bit more, perhaps a bigger picture view of everything. So what's one of the big implications about all this? Well, the observation that life responds to changes in magnetic fields, suggests that actually life is electrical, which means that potentially quantum mechanics can provide insight into health and disease. And I think we're getting familiar with this, and certainly from the electric field thing, this idea that life is electric goes back a couple of hundred years, and back to Galvani, and who was playing around with his frog's legs. And he was actually in competition with Volta, who was suggesting that electricity is all to do with physics. Well, actually, we could all say that biology is all to do. Electricity could be due to biology as well. We've also moved on to, of course, to quantum mechanics. One of the things that we're very interested in, of course, in this area is spin. And if we look through the history, the origins of spin go back into the sort of 1920s, but an experiment by Stern Gallack, who discovered something they weren't quite sure how to explain, but that Erlenbeck and Goudsmit came up with this theory of spin, which was later developed and fully developed by both Pauli and Dirac. So linking kind of the electricity into the spin idea. But of course, the big question is why is life electrical? Well, actually, when we start looking at as I always do, is go back to the origins of this idea. It's about the descent of the electron, and of course we know all about electrons. And if one of the starting places for a lot of this, of course, is to go back. How did life start? Now, there are clearly other alternative hypotheses, but one of the favourites at the moment is this alkaline thermal vent idea. And of course, the theory here is that we had a large energy gradient, and in particular, we had the upwelling of hydrogen meeting an acidic sea, and this could generated like autocatalytic networks. And these seem to fall out of the idea of thermodynamics and quantum mechanics. And this led to the evolution of electron transport chains, and in particular these various molecules here like FMN, NAD, which transfer electrons. And water is particularly important in all of this. And of course, we see this today in electron transport chains like this one shown here, which is the standard one, where electrons flow and we generate a proton gradient. And so we have this flow of electrons generating this what we see in all life today. But of course, the big question, and the one I'm trying to attempt to answer, but it's a very gentle answer, is why do electrons move at all? Well, this is another way of looking at this. And one of the things is, if we go back right to the beginning of time, and again, other theories are available, we had something like the Big Bang. And when we point our telescopes into the universe, we now see that actually the universe is full of prebiotic molecules and water. And this is something which wasn't always thought to be the case. So we're seeing evidence of things like nucleotides, polyaromatic compounds, and from Bennu and places and asteroids, we're now seeing amino acids. So the ingredients for life were all there. So what triggered it off? Well, one of these ideas is that because of this energy gradient, we had a flow of energy, and it seems from the laws of thermodynamics and quantum mechanics, seems to generate self-organizing structures. And this is the ideas of Ilya Sinayskiy, but this seems to where life actually started. But what's interesting about some of these structures, they're trying to dissipate the energy potential, but they're also under the process of natural selection. So even Darwin probably would have understood partly what's going on here. Anyway, at some point, 4 billion years ago, we ended up with some kind of proto-life or life, and we call this a small contra of life, which is an autocatalytic network. And this took on board energy and information, and in fact exported entropy. And if we have a quick tour through the history of life on Earth, some very interesting studies suggesting that the Earth was actually a water world very early on, but then may have gone purple. And again, this falls out of the idea that some of the very earliest autocatalytic networks were generating things like isoprenoids. So actually, phototrophy, the generation of a proton gradient, could have happened very early on, and it absorbed in the green, which of course explains why everything's green nowadays, because photosynthesis tends to work on the blue and the red end. But then we got the big event in history, which we've all talked about many times, is the origins of mitochondria and of course eukaryotic cells. And again, science is moving really, really quickly on this, because these things called archaea are turning out to be far more important, far more interesting than anybody ever imagined, because they have a very complex prokaryotic cells. So it's another order compared to the bacteria. Anyway, these got together and we had the modern eukaryote, and these things started to multiply and evolve. Eventually, we ended up with humans on the planet, and we are churning through huge amounts of energy now. We're getting through trillions of gallons of oil every year. We've developed fusion and fission rockets, and now we're into AI. And this has led to this whole idea, of course, that life is just a way of accelerating the heat death of the universe. Though other theories are available, but it seems to be quite an interesting one. But the implications from this are pretty big. Now, Of course, if we move charge, quantum mechanics tells us we get fields. And if we go back a couple of 150 years or so, one of the big theories at the time, on the turn of the 19th to 20th century, and this is before we knew anything about DNA, was that life actually, the shape of life and embryogenesis and regeneration was actually down to some kind of morphogenetic field. And this character, I think we've talked about many, many times before, is Alexander Gurwitch. And he was a real proponent of this. And he was actually searching for the field, as it were, and when he discovered his onion biophotons. And of course, this led to all sorts of things. But more modern day, we've got this. Mike Levine has been doing some fantastic science showing that bioelectric fields are indeed extremely important in all of this. This is through to ion channels and the movement of ions. And then we move on to the idea of electrodynamics, which many people are involved with, including Mikkel Sifra, who's always spoken with us before. And so really, when we start to think about life, we had to think about the movement of charge and fields and the sort of yin and yang of fields and molecules. We know everybody's brought up in biochemistry and medicine. It's just about pharmacology and molecules. But actually, the fields are just as important. And of course, this brings us back to electron spin and magnetism as well. Now, of course, this is all very exciting, but, if we have a theory on morphogenetics, electric fields, when did it start? Well, we had a stab at this a while back. And if we go back to our old thermal vents again, one of the things you can argue is that they were moving electrons and protons around, which would have generated fields. Now, the other piece of quantum mechanics that may have come into this, of course, is this idea of ordered water and tunneling. And all of these things could have come together at some point to develop like an autocatalytic network where fields were very important. And this could have led to, like we suggested, the first quantum of life. But of course, it suggests that what defines self from non-self is actually an electric field. And this is one of the very first things that happened. And there are all sorts of interesting things that fall out of this. One of the ones is that actually cooperation was something which happened very early in evolution. We're often taught that it was every cell for itself in early evolution back 3.5–4 billion years ago. But there's an alternative view that actually it was all about cooperation and a mingling of different fields. And when we start to apply this idea of fields and this dissipative self-organization, we start to get towards some understanding of things like cancer, say the role of mitochondria, and how, for instance, say exercise seems to be anti-cancer, while overeating and under-exercising tends to be pushing towards cancer. I haven't got time to go into this, but this all begins to link together quite nicely. And of course, there's always about the proof of concept here. And again, this is all life has an electron of some description. And again, people say, oh, yes, well, we've discovered this. Well, and of course, axons and things, they generate pendulum. We know the brain's got its electric field, and we know the heart has. But when you start going back through time and then looking at prokaryotes, what we've discovered, and this is back to this cooperative idea, we are finding everywhere now these colonies of prokaryotes, and this is consortia, as they call them, of archaea and various bacteria. They're sharing electrons. Not only they're sharing electrons, but they're also showing electric fields. But of course, if you look at the most basic cell, like a bacterium, it's all about its movement of ions across its membrane, which defines its electric field. And we can start to see this in other areas. We look at, of course, things like cancer, and there's all these theories around, and Martin was talking about this, the role of electric fields in cancer. We look at microtubules. There's loads of evidence now this is important. Of course, even plants have their own electron, and they signal through some kind of electrical transfer. But there's been bigger theories on this now. When we start to look at, say, forests and the Amazon, life is actually all connected. And so this idea that trees are talking to fungus, which are talking to other trees, this could all be electrical as well as chemical. So we come back to this idea of magnetic fields. And again, we have to start thinking about, well, okay, it's all very well about magnetic fields, but why is it important? Well, one of the ideas is that Earth, the early Earth, the Hadean Earth, and this is sort of getting on for 4.5 billion years ago, as it cooled, it started to generate its own magnetic fields. We don't really have evidence of this, but until we get to about 3.5 billion years ago. But there is evidence there was a magnetic field, suggesting that for most of the evolution of life, there was a magnetic field, geomagnetic field. And we know it did change direction occasionally, and occasionally it got weaker and stronger. But overall, there's always been there. And of course, when we start to think about life evolving, it would have come what we call canalized to these conditions. It would have evolved in what we call it would have developed its own magnetic flight envelope, i.e. it was dependent on its envelope to function correctly. And this comes from if we start thinking about spin and electron transfer. And of course, if we flip this over to we get a curve like this, we get the opposite of a U-shaped curve, we've got a Goldilocks zone. And what seems to be the case for all biology, that it operates in this area. And so you can argue that if we start to change the fields, we get too little or even too great, we might change, say, the flow of electrons, which of course could induce reactive oxygen. And of course, it's not just about the strength, but it's also the direction and modulation, and as has been discussed before. So where is the data? Well, I think I don't need to tell anybody, we've got shed loads of data on this now. If we start looking through the literature, there are literally hundreds and hundreds of studies showing that, yes, magnetic fields do affect biology. This goes from obviously the bird navigation, but much more fundamental things turned due to metabolism. And this is some of these reviews here, like from the Russians, from the Saramov, and the Chinese in particular. I've been looking at this for a very long time. And one of the key targets seems to be mitochondria, which you would expect, because of course it's where the biggest flow of electrons is probably happening all the time. And of course, it isn't just about radical pairs, and we haven't got time to talk about this, but about magnetite. And this is paper by Peter Hoare is talking about the fact that magnetite and radical pairs could work together. And of course, magnetite was right there at the beginning of life. So we've got to think about these things. So why is all this important? Well, it tends to validate quantum biology as being an approach. It also potentially validates hormesis, this idea of what doesn't kill you makes you stronger. But also, if we think about the implications, it could take us all the way back to the origins of life. We could be seeing an echo of this today. Clearly, we know about the clinical applications of all of this. And as you said, as Martin and others have talked about this. But we've also been very interested in this, of course, from the space angle too. So just to sort of summarize this bit, the main implications are, this idea of the vital magnetofield. Sorry, that's a pretty terrible word. But of course, if you've seen this diagram before, life has certainly canalized the magnetic fields. But what we've got to also bear in mind here when we're studying this, it's also canalized, it has evolved under a constant gravity, under a shifting light spectrums, gases, temperature, radiation, even movement is particularly important in all of this. And diet, and we talk about diet as providing energy, but it also provides lots of compounds which are redox active, polyphenols. And a lot of polyphenols, they say they're antioxidant, they're also oxidants, and they're plant stress compounds. So all these things goes together. So when we look at how life is kind of canalized, it's like a nested series of all of these things are important. And we can look at this again with our Goldilocks zone, seeing the same thing. But clearly, one of the most important aspects of all of this, if we're talking about the descent of the electron, is actually this redox balance. Reduction oxidation has to be absolutely balanced. And when homeostasis is disrupted, usually one of the effects is a change in redox, and the system picks this up. Now, of course, one of the interesting things about this is where the hormesis comes in, this idea certainly of mitohormesis. What we find is that when cells are stressed, life is stressed, there generally seems to be a common reactive pathway. There are variations, but quite often they're similar enough that one stress from one thing can induce adaptation and protection against another. And we raised this possibility in this idea about COVID, and we predicted, and as many others actually predicted, that when COVID struck, the most liable populations to get ill were those who are metabolically not very healthy. And one of the ways of defining metabolic health is actually mitochondrial reserve, because mitochondria don't just produce energy, they also act as reactive oxygen sinks. So this ability to manage oxidants and stress is actually really, very critical. And so when we take this on board with the magnetic fields, we can start to say, well, actually, depending on where you're starting, magnetic fields or changes in magnetic fields can both be damaging, for instance, in a hypomagnetic, but it can also be restorative. Because if you're in, say, an inflammatory state, you might be able to nudge the system back towards a central point, like an attractor in the middle, to restore, say, suppress inflammation. And so I was going to talk about the space study. Well, one of the ideas here is that, of course, if we're talking about space, what will hypermagnetic fields do? Will they accelerate the aging process? And this is particularly important, as Massimo was talking about, beyond low Earth orbit, where we run out of magnetic fields. And will this compound other problems like the lack of gravity? Will we have to look at spacecraft design? If we start to look at, say, this diagram here, and this is we published this earlier in the year. If you look at where we are now on Earth, we're subject to this gravity, magnetic fields, circadian rhythms, photons, gases, and radiation, and we evolved in this sweet spot. And these can vary a bit, but we can adapt to it. Go into space, and all bets are off. There's no weight. We've got the magnetic fields can be different. Our circadian rhythms are all over the place. If you're on the ISS, you've got 16 days a day. Your photonic environment could be all over the place. Your gases are all over the place, and you increase radiation. All of these things start to become really quite important. And has been discussed before, is accelerated aging in space a thing? Well, the answer is many very clever people say it is, and certainly the data is beginning to support this. Space in a spacecraft is a good model for accelerated aging. And this is something I think really does need to be taken on board quite seriously. And so these are just some of the review articles that talk about this. In particular, of course, we're looking at, say, mitochondria, and we know they get very bad. They start to dysfunction in space. When we think about the radiation, and just come back to the most fundamental point about it all here, is that we live on this little green dot here, or blue dot as we like to call it, but because of our magnetic field, we are protected from very high levels of radiation coming in various variety of forms, certainly from the sun, solar particles, and mostly protons and other things, but galactic cosmic rays. And these things are very nasty. These are high energy particles. And they're generated in things like supernovae and things. They may take millions of years to get here, but these are things like, the nuclei of say, iron or carbon, and they are traveling at relativistic speeds. These are going fast. They contain huge amounts of energy. We don't get many on Earth, but out in space we do. These things are really dangerous, and most people studying space at the moment haven't come up with a solution to this yet. Apart from the possibility, and this is something which has come out quite recently, although it's an old science fiction idea, we generate our own electric magnetic field on a spacecraft or on a moon base. But of course, the principal question here is, we may only need a field strength of 300 nanotesla to 1 microtesla. But what happens inside the field? Do we have to protect the astronauts from a change in the electromagnetic field? So these things it's quite big implications. And this is, again, this idea, this is from the Martian. But when we start to look at designing a spacecraft, artificial gravity keeps coming back, as I'm sure Jack would agree with me on this one, as being really important. But of course, this enhances the ability of exercise to work. But then, of course, we've got to think about the right light spectrum in there. And of course, light can affect molecules which absorb, molecules in the electron transport chain and therefore spin. We have diet. This could be very important. We also have to think about the shielding, as you mentioned, of course, even the right oxygen to carbon dioxide balance. And in space stations at the moment, we have too much carbon dioxide, which could be quite damaging. So how do we mitigate all of this? This is quite a big problem. And so I'll kind of finish on this one. And, we've mentioned this before, but, there's this great push at the moment to send people off to Mars. But what the theory seems to be telling us is that if we spend 6 months in a spacecraft with no gravity, with no magnetic field and wrong light and everything, we may not be very well at all. And the hypermagnetic fields being potentially quite a big component of this. And in particular, of course, when we start to think about how things adapt, humans won't adapt very well at all, or don't appear to. We don't have any data, but the suggestions they won't. Your microbiota, on the other hand, your bugs in your gut and everywhere else will evolve very quickly. So what arrives on Mars? Is it us or our bacteria, or an evolved version of that? And with that, I shall finish. And we'll move on to the round table. Thank you very much.